Hello, my name is Miwa, and today I'll be talking about the structure of intercellular junctions. Before actually getting into the topic, let me explain how the highlights in my slides work. Red highlights are for terminologies that you must know for the exam. Orange is for the terms recommended to know for acing the exam, though not essential. And greens is for terms that you do not need to memorize, but just good to keep in mind. All right, let's dive in. So what exactly is an intercellular junction? It's a structure that allows communication or adhesion between cells, or between a cell and the extracellular matrix. When we talk specifically about junctions between cells, we refer to them as the junctional complex. There are 7 major types of junctions and 1 minor one. These include tight junction, adherence junction, desmosome, gap junction, hemidesmosomes, and focal adherence, and lastly, fascia adherence. Now let's go into each one in detail. Tight junction. Tight junctions form continuous seals between cells, which is why they are part of the junctional complex. Their main functions are to isolate the apical part of the cell and to create a strong adhesion between adjacent cells. Tight junctions are made up of various proteins, including claudin, occludin, JAM, and zeoproteins. The associated cytoskeleton is actin. Where can tight junctions be found? Examples include the blood-testis barrier and the blood-brain barrier, or BBB, and more. The blood-testis barrier is located in the seminiferous tubules in the testis. Why does it need a tight junction? Because sperms are recognized as non-self, and the tight junctions help prevent autoimmune reactions. In the brain, the BBB restricts unnecessary substance exchange across blood vessel walls. Next, adherence junction. Just like tight junction, adherence junctions form continuous seals between cells, making them part of the junctional complex. Adherence junctions are composed of proteins such as cadherin, P120, and catenin, and importantly, calcium ions. The calcium ions help ensure adhesion between the right type of the cells. The associated cytoskeleton is also actin. Where are adherence junctions found? Examples include the epithelia of the endometrium in the uterus and the outer limiting layer in the retina. The left scheme represents the endometrium and the right one, the retina. In the endometrium, they allow for the invasion of the blastocysts during implantation. Next, desmosome. Unlike the previous two, desmosomes provide spot-like adhesions between cells. Desmosomes are made up of intracellular components, such as desmoplakin and plakoglobin, and extracellular components like desmoglein and desmocolin, which belong to the cadherin family. The associated cytoskeleton is intermediate filaments. I've listed some of the important intermediate filaments found in different cells. Next, gap junction. Gap junctions act like tunnels between cells, allowing for the direct passage of small molecules. They are made up of connexins, and when 6 connexin molecules aggregate, they form a connexin. The primary function of gap junctions is to allow the transfer of small molecules like ions, sugars, and amino acids between cells. Where can gap junctions be found? Examples include intercalated discs, in cardiomyocytes, especially on the longitudinal side, electrical synapses, and the canaliculi of osteocytes. Next, hemidesmosome. Hemidesmosomes connect a cell to the extracellular matrix. They contain integrin as the extracellular component, unlike desmosomes, which contain cadherins. The associated cytoskeleton here is intermediate filaments. Next, focal adherens. Focal adherens attach a cell to the extracellular matrix. They are structurally similar to hemidesmosomes, but they are smaller in size and more numerous. The associated cytoskeleton is actin, which also differs from hemidesmosomes. Finally, fascia adherens. While it is a junctional complex, it's not usually mentioned in textbooks, so I'll just briefly explain it. It is functionally and structurally similar to adherens junctions, and is found in the transverse components of intercalated discs in cardiomyocytes. That's it for the major points about intercellular junctions. I hope you found this interesting.